Okay, so a number of you guys gave the feedback in our mid-semester check-in that you would prefer a greater number of shorter video lectures rather than one really long video lecture. So this is my attempt to incorporate that feedback. There's going to be a set of three of these videos that go in this sort of playlist for mitosis, and this is the first of those videos. So in this video, we're going to be talking about DNA and chromosomes. And the reason that we're going to be thinking about this is because DNA is a super important molecule, and it is the only family of macromolecules that we haven't covered yet. So we've talked about lipids, we've talked about carbohydrates, and we've talked about proteins. And the last major family of macromolecules is nucleic acid, which would include the molecule DNA. So DNA is a molecule that contains all of the instructions for how to build proteins for an organism. And now that we've covered what proteins are and what they do, hopefully you guys all understand that proteins are super, super important, right? They do so many things. I mean, the enzyme bit is just one piece of what proteins do. They are fundamental for a cell to be able to function. So, if a cell is going to make proteins, which is virtually all cells, that cell needs to have DNA to provide the instructions for those proteins. And you might be wondering why we're talking about DNA when I said this was a mitosis playlist. But the reason that we're starting with this one is that as we're thinking about a cell's life cycle or what the different stages that a cell goes through, um, starting when it's formed and as it continues to sort of live on, the DNA in that cell is going to look different depending on the stage that the cell is in. So we're going to be paying particularly close attention to what the DNA looks like at different stages of a cell's life cycle. And sort of knowing what characteristics to look for when we're thinking about DNA will be, I think, helpful as we're thinking about the cell cycle and mitosis. So, there are two main questions that I want you to ask yourself when you're looking at DNA or looking at the DNA in a particular stage of the cell cycle. So the first question is it packed up tightly or is it unwound and loose? So these are the two main forms that DNA can come in. So if the DNA is packed up super tightly, then we call that a chromosome. So this familiar example of like the X-shaped thing that usually that's how you draw DNA. That's we call this form of DNA a chromosome. But DNA can also be found sort of like just like loosey goosey. People compare it to a ball of spaghetti, just sort of loose and unwound. I'm repeating myself. But it's sort of just like a tangled up mess, kind of like an unraveled ball of string or something like that. So when DNA is in this form, we call the DNA chromatin if it's in this unwound form. So that's the first question. Is it unwound and loose, in which case we're calling it chromatin, or is it in this super compact X-shaped form, or or it could even just be one side of this X, in which case we're calling it a chromosome. So packed up tightly chromosome, unwound chromatin. And then the second question I want you to ask is the DNA a single copy or is each strand of DNA replicated or duplicated? So this is the other big difference for how DNA can look. Um, in some parts of a cell's life cycle, it exists as just a single piece. So this is one molecule of DNA, and each part along this molecule of DNA is unique. There's no parts of it that are copied or duplicated. But at certain stages of a cell's life cycle, the DNA gets copied so that we have two exactly identical copies of the same information. So this makes sense when we're thinking about the fact that cells are going to need to divide, and that's what this video lecture is sort of all about. If a cell is going to divide, it's going to need to give its genetic information to both of its offspring cells, right? So at a certain point in the cell's life cycle, it's going to divide or copy its DNA so that when it divides, both of its daughter cells can get the full amount of genetic information. So, our two questions: DNA, is it packed up tightly or is it unwound? And does it exist as a single copy, or is it duplicated or replicated? So let's explore both of these in slightly more detail. Okay, so this is just to reiterate the point that DNA can be loosely or densely packed. And this is a diagram showing that sort of canonical looking chromosome that you maybe are used to imagining when you think about what a chromosome looks like. And what you can see is that making up this chromosome is this super um, coiled up DNA that's coiled around these proteins called histones. And then the histone strands are coiled upon themselves and it's coiled up even more. We call DNA super coiled when it's in the chromosome form, and that's because it really tightly packs all of it up. And then when it's more unwound and sort of unspooled, that's the chromatin form. And you can think about this in the sense of thinking about what the cell will need to be doing with the DNA, and that will inform whether the DNA is loosely or densely packed. So it's almost like when the cell is just hanging out and it needs the DNA to be accessible, then you don't want it to be sort of tightly packed away, right? So if you think about your clothes, if you want them to be accessible to you in your day-to-day life, like you want to put on whatever shirt you want, you're not going to have each and every shirt that you own packed up into a tiny little suitcase, right? Like that's not practical. So when the cell needs access to its genes, it's going to have it in this more unwound chromosome form, which is more chromatin, unwound chromatin form, which is more open. But if the cell needs to be moving around its DNA, like if it's shipping it off to two different daughter cells and needs to sort of keep track of all of the pieces that it has, then it's going to move into this condensed, densely packed chromosome form. And that just lets the cell keep track of where all of the pieces of DNA are and lets it move the DNA around more efficiently. So we've got chromatin and chromosomes as our two main forms that DNA can take depending on its packing structure. Okay, and then the other question that we were thinking about was whether DNA is going to be replicated or unreplicated. So here we're showing a single unreplicated chromosome. And I was saying that before a cell divides, it's going to need to copy its DNA so that each daughter cell, each of the resulting, if we're making two cells out of one, we're going to have, we're doubling the number of cells we have. So we also need to double our genetic material. So at certain points, cells are going to copy their chromosomes and will end up with what's called a replicated chromosome. So this X form of a chromosome that you're used to seeing, and this is a picture of an actual chromosome, has these two sides to it. And this is one chromosome, and then this is an exact copy of the other side. And the region where these two chromosomes are held together, we call that a centromere. So centromeres are just the regions in the middle that hold these two copies of the chromosome together. We have more grammar. We're not done yet. So when we're thinking about what to call these copies, we call them chromatids. So instead of chromosomes, they're chromatids when they're in the copied form. And specifically, if we're thinking about two copies of the exact same chromosome, we're going to call them sister chromatids. So these sister chromatids are exact copies of one another. They're exactly the same. So here is this diagram form. You can see that these two arms are sister chromatids, two copies of the same chromosome. And so then when they get pulled apart and they're going into the daughter cells, now they're not chromatids anymore. They become their own. They mature into chromosomes. We call them chromosomes now. So they go from chromatids to chromosomes once they're on their own again. So chromosome gets replicated to become sister chromatids held together at the centromere, and then when they're separated, they become chromosomes again. So replicated versus unreplicated. That's the other big question we have to ask ourselves. Okay, that's it for this part one for this first video of our mitosis series.